Hello students, welcome to my channel D Biology. In today's video, we are going to learn about the light reaction. Yes, the light reaction which is of two types. One is cyclic photophosphorylation and another one is non-cyclic photophosphorylation. But before we start with this two types of light reaction, what things we need to know is what is light reaction basically, what is the site of this light reaction, what are the parts involved in this light reaction, because once this basic is clear, then we can easily understand the two types of photophosphorylations or two types of light reactions we have. So, this light reaction is the first part of photosynthesis. Photosynthesis is basically divided into two types, light reaction and dark reaction. Why the names are given? I'll tell you. Light reaction occurs in presence of light. Means if light is not there, this light reaction is not possible. Now, dark reaction, the other part of photosynthesis, doesn't mean that it does not require light. It is indirectly dependent on light. The dark reaction is indirectly dependent on light, whereas this light reaction is directly dependent on the light. Hence, the names are given. Dark reaction can be considered as a misnomer. Clear? Now, in the presence of light, they produce the products like ATP, NADPH2, oxygen. I'll tell you in which process what are the products formed. So what are the products formed in the light reaction? Basically ATP, NADPH2 and oxygen. Out of these three products like ATP and NADPH2, these products are actually used by the dark reaction. These products will be used by dark reaction, means now they will be entering the dark reaction. Clear? So the products of light reaction are used in dark reaction. Hence we say that dark reaction is indirectly dependent on light reaction, because if these products of light reaction are not provided, dark reaction is not possible. So even if we cause complete darkness, but we provide them these ATP and NADPH2 to the plant, dark reaction is possible. So I hope now the names and the reasons of their names are clear to you. Now coming to the light reaction again, this light reaction has been given few more names. One is Hill reaction. Now Hill reaction is on the basis of the name of scientist who gave this light reaction concept. Then it is also known as electron transport chain, because here we'll see that the complete system, the complete action is actually done for the electron. Everything is about the electron, from where it is going to where. So here we call it as electron transport chain, another name, electron transport. Once the basic is clear, it is very simple topic, electron transport chain. So remember, these are the two names of the light reaction. Why these names are given? It is clear. Now, it occurs in presence of light, as I told you. Here, what happens? What is the site? So guys, what happens? Specific structures are present in the chloroplast. Specific structures which are named as thylakoids. Thylakoids are present in the chloroplast in which this light reaction is going to occur. I'll explain you where these thylakoids are found and everything. So, in these thylakoids, pigments are present. Those pigments trap the photons of the light and then they help in the ejection of electrons, causing this complete electron transport chain. So let me first show you what these thylakoids are, as you have already seen the thylakoids in your cell biology unit. But again, we'll have just a quick revision about the thylakoids, since we are going to study here the role of thylakoids. So guys, this is one chloroplast. This is, let's say, the outer membrane of the chloroplast. Remember, chloroplast is double membranous structure, right? So this is the inner membrane which I'm drawing now. Inside this inner membrane, there are some structures which are present in a stacked form. Like this is one disc shape, this is one flattened structure. I'll be drawing this over here. This is one flattened structure, right? Now, if I put more flattened structures in the form of sac, look how it looks like. It looks like a stacked form of these flattened structures like this. So this one flattened structure is actually one thylakoid, whereas this complete stack is known as grana. It is known as grana. Whereas if we draw a single thylakoid, like one more stack is here, okay, this is also again a complete grana. These two granas are further joined by a single flattened structure. The single flattened structure is known as the thylakoid. So thylakoids stack upon each other to form a structure known as grana, thylakoid, not S. Thylakoid, right? So we can have more than like so many thylakoids and grana within the chloroplast. The fluid matrix which is present in the remaining part of the chloroplast is known as stroma. Clear up to here? Now, other than this, we are going to see one thylakoid now. As I told you, this is one thylakoid. If this thylakoid is stacked with a number of thylakoids like this, then this complete structure formed is known as grana, whereas each one is known as one thylakoid. Clear? Now, if I draw a single thylakoid inside over here, I'll draw it over here. Then, nothing much is there for us to learn, just you need to remember the things. Easily they can be remembered. This is one thylakoid, okay? Now, this thylakoid I'm drawing, I'm just coloring one of the thylakoids. Just this part I've drawn. Now, this complete part is the thylakoid membrane. What it is? It is thylakoid membrane. Because later we'll be talking about these sites also. So we need to have knowledge about what is thylakoid membrane, what is the lumen. Okay, now the space which is enclosed within the thylakoid membrane, like this one, this complete space, this is basically the lumen. This complete space is what? This complete space is lumen, which is enclosed by the thylakoid membrane. Now again, if we come towards the thylakoid membrane, then this complete is thylakoid membrane, but this is the inner thylakoid membrane, inner part of the thylakoid membrane, and this is the outer part of the thylakoid membrane. Okay, suppose this is one complete membrane. This is inner, this is outer membrane. It is single membrane, but we are talking about the inner part and outer part. It does not have two membranes. Usually we do get confused, are they having two membranes or what? No. They have one membrane, but it is thick, so we talk about the inner part of the membrane and outer part of the membrane. Clear? Now, so let's label this part. This is outer thylakoid membrane, OTM, and this is ITM, inner thylakoid membrane. Now, surrounding to this thylakoid, as you can see, here we had this thylakoid. Surrounding to this thylakoid, what is present? Of course, we have the stroma. So, in the surroundings of this thylakoid also, which we draw separately, outside this outer thylakoid membrane, what will be present? Stroma will be present. Clear? So, this is one main point which you need to remember about the structure of a thylakoid, because when we will study about the two types of light reaction, we'll definitely talk about the inner part, lumen, thylakoid membrane, stroma, everything. Because from that part, important questions are framed, and a number of times they are asked in the exams. Clear? So this is about the structure of the thylakoid, where this light reaction will take place. Apart from the structure, we need to have knowledge about the pigments. Those pigments or molecules which are trapping the light energy, which are trapping the photons. So let's see what these pigments are or what these molecules are. So we know that the photosynthesis basically takes place in the presence of which pigment? Chlorophyll. We have been saying this from our junior classes that chlorophyll containing parts, green parts of the plants, they do photosynthesis. But what does chlorophyll do? Is it a single kind of molecule? Is it a number of types of molecule? What it is? So guys, to understand this, we need to understand or have knowledge about the light harvesting complex. The next point is light harvesting complex. Harvesting means what? Harvesting means one who get the results or one who traps, right? So here we have a complex of pigments which traps the photons of the light to eject the electron. So how this complete system goes, I'll explain you this point now. So basically for performing the photosystem's first part, that is light reaction, individual pigments are not performing the function, but they are performing functions in a complex structure. Clear? Around 230 molecules. How many number of molecules? Around 230 molecules of chlorophyll. 230 chlorophyll molecules are going to form one quantazome, and that particular structure or this light harvesting complex will trap the photons of the light. So I'll show you how they look. This is one light harvesting complex which I'm going to draw over here for you. So many pigments are there. Apart from chlorophyll also, like 230 molecules of chlorophyll on average are present. But other than the chlorophyll molecules also, some other pigment molecules like carotenoids, xanthophylls, they are also present here for trapping the photons. So this are all the pigments are present here. Okay. Chlorophyll A, chlorophyll B, or if any other type of chlorophyll is there in the plant, it is also present here. These are the pigments which are present in a form of a complex that is known as light harvesting complex. Now, you know that when we use this dish TV and all, we get a antenna on our terrace. That antenna gets the signals, and then we get the things on our television. In the same way, the light harvesting complex has got some antenna molecules. These antenna molecules are not trapping any signal. Of course, they are not trapping any signal, but they are trapping the photons of the light. So let me complete this part of the photons. Just a minute. Now, in this light harvesting complex, there is one main center, one main pigment or molecule of chlorophyll A. This chlorophyll A main molecule is known as reaction center. Reaction center. Now, all other molecules which are present in the complex, these molecules are known as the receptors or antenna molecules. Now, what happens basically, we'll use blue color to show the photons of the light. So, suppose these light photons are falling here. So, the main pigment for photosynthesis or for light reaction is chlorophyll A, but chlorophyll A itself cannot take all the photons necessary for performing the light reaction. Just like if we talk about the Our school principal, let's say, our school principal is the main one, right? But he cannot control all the activities, all small tasks going on within all the classes. So what he does, he gives powers to different teachers. But all those teachers finally they have to inform, they have to give reports to whom? They have to give reports to the principal. Just like that, yes, it is an interesting example, right? We all can relate because all of our complaints go to principals by our teachers, right? So what happens, these all red molecules are basically the teachers which are sending the reports, these light photons. What this blue color is, this is the light photons. They are trapping the light photons because this main or the reaction center, the principal cannot trap all the light photons. So the pigment molecules, accessory pigment molecules will trap the light photons and will transfer these light photons through these pigments, and finally these photons energy will be sent to chlorophyll A. Now this transfer of energy is known as resonance transfer. Resonance transfer. Here, suppose we do something mischievous in the school. Our school principal will not get the news directly, but the teachers will send the news to the principal. So, our complaint is the light photons. Then these red molecule of the pigments are the teachers. They are just gossiping and transferring the complaint about the student to this principal, which is the reaction center. Clear? So all the teachers, all the pigment molecules have to gather light photons and send to the chlorophyll A, the reaction center, through the process of resonance transfer. Resonance transfer is the process where the electron energy, the photon's energy is sent to the chlorophyll A. Once this chlorophyll A gets the energy, now it will eject the electron. Means the punishment is given by the principal. Very easy to remember. Okay, so light photons sent to the chlorophyll A, that is reaction center. From that reaction center, electrons will be ejected out. Now, on the basis of two types of, means we have two types of chlorophyll A, that is chlorophyll A 700 and another one is chlorophyll A 680. So on the basis of a wavelength which this chlorophyll absorbs, the reaction center chlorophyll A absorbs, we have got two types of photosystems. Okay, one is photosystem 1, which has chlorophyll A 700. It is photosystem 1, and another one is photosystem 2 that has chlorophyll A, which absorbs the photons of light wavelength 680 nanometer. So we need to have a small amount of knowledge about these photosystems actually, because one of them is absorbing the wavelength above 680 nanometer and one is absorbing the wavelength below 680 nanometer. And on the basis of these photosystems only, where they are functional, where they are non-functional, we have got two types of light reaction, that is cyclic and non-cyclic photophosphorylation. So this light harvesting complex is completely done. When I say light is falling and electron is ejected, this complete process will be done over there. I'll not take this portion again when I'll discuss the two types of photophosphorylations. Okay, so let's move to the next part where we are going to discuss about the two types of photosystems. So first we have got photosystem 1. And then we have photosystem 2. We'll study these two photosystems in a comparative form so that you won't get confused at all. You won't have to mug up anything. You just have to understand and remember. So coming to the first point about the photosystems. Photosystem 1 has reaction center as chlorophyll A. What is its reaction center? Reaction center is chlorophyll A, which is absorbing the light of what wavelength? 700 wavelength, right? So chlorophyll A 700 is the reaction center, whereas here in photosystem 2, chlorophyll A 680 is the reaction center for photosystem 2. Now, we are going to link these photosystems with the thylakoids. So I'll draw the thylakoids on side over here. This is one thylakoid, but if I stack a number of thylakoids together, what they are going to form? They are going to form the grana. So the second portion has a grana with number of thylakoids. Clear? Now, in this grana, the portion means this portion where the two thylakoids are in contact with each other, this portion is known as oppressed region. Oppressed region of thylakoid, oppressed region, and the remaining part which is not in contact with the another thylakoid, like this complete surface, it is not in contact with another thylakoid, so we call this surface, means this complete portion, as non-oppressed region. Non-oppressed region. Clear? So, photosystem 1 is found in non-oppressed region, whereas photosystem 2 is found in oppressed region. So guys, can you conclude one point over here, that photosystem 1 will be present in both thylakoid, individual thylakoid, and in complete grana also, but photosystem 2, which is present only on the oppressed part, cannot be found in the individual thylakoids. It is found only in the oppressed region of the grana. Clear? So next point is found in both thylakoid. Thylakoid means individual thylakoid and grana. Now, that individual thylakoids are known as stromal thylakoids. Stromal thylakoids or stromal lamellae, and the thylakoids of grana are known as grana thylakoid, so that you won't get confused. Thylakoids. Clear? Coming to photosystem 2, they just have, they are just present on granule thylakoids. Granule thylakoids. I hope till here no doubts are there. Now, photosystem 1 is going to participate in both the types of light reaction, that is cyclic and non-cyclic. This you just remember here. We'll understand, we'll see their functions when we'll study the two types of photophosphorylations. So, present in both cyclic and non-cyclic. Sorry, it is non-cyclic photophosphorylations. Photophosphorylation. Whereas this photosystem 2 is found only in the non-cyclic photophosphorylation. Only in non-cyclic photophosphorylation. Phosphorylation. Okay. So I'll not explain here what is the cyclic and non-cyclic terms, but I'll explain what is this photophosphorylation. You get to study in plant physiology two terms, oxidative photophosphorylation and photophosphorylation. Photo means what? Light. Phosphorylation means what? ATP formation. So photophosphorylation means formation of ATP in the presence of light. That is seen during the light reaction. Coming to oxidative photophosphorylation, a kind of ATP formation by oxidation process that we see in the chapter respiration. Clear? So I hope you won't get confused between the two terms from now. Now, coming to these photosystems, who gave the concept of these two kinds of photosystems? So here we get another important point, that concept of two photosystems. Two photosystems means the one who gave the idea that we have two photosystems going on within the plant, within the same plant, we have two photosystems. It was given by two scientists, that is Immerson and Arnold, and they worked on an algae that is known as chlorella to give this concept. Chlorella. Clear? Now, what is the concept here? The concept is that PS1 is used in both the photophosphorylations, PS2 is the sun, it is used in only non-cyclic photophosphorylations. But Immersion had more work on this, and we get to study two more points regarding this complete process. One is red drop, and another one is immersion effect. Immersion effect. Students get afraid of this immersion effect and red drop, but it is very, very easy to remember. Immersion effect is also known as enhancement effect. Enhancement effect. It is very, very easy concepts. Now, when these people were working, they saw that when they used just wavelength of light above 680 nanometer, the outcomes of photosynthesis was very low as compared to the normal production. So they saw a drop in the production. They gave the term red drop. It is seen when the wavelength of the light is above 680 nanometer. And when 680 nanometer above wavelength is given, only PS1 remains functional. PS2 becomes non-functional. Clear? That's how they gave these concepts of photosystems. Coming to the second concept, or the immersion effect, they said that when we use wavelength of light both above 680 nanometer and below 680 nanometer, we get to see a huge increase in the production, in the productivity of the plants. This huge increase, enhancement, is known as immersion effect or enhancement effect. It is done because both the photosystems are active. When we give the wavelength of light above 680 nanometer, who becomes active by this one? PS1 becomes active. And if we give below 680 nanometer together, then who becomes active because of this below 680 nanometer wavelength? Photosystem 2. Photosystem 2 becomes active. So this is all about the two photosystems, red drop, immersion effect and effect. Nothing difficult is here. That's what I think, and of course I'm right? So here we are done with the concept about the photosystems, and now we are here with our main part, that is the two types of light reaction. So what are the two types I told you? The two types of light reaction are cyclic photophosphorylation and non-cyclic photophosphorylation. Means one process where the ATP is formed by the light when a complete cycle of electron is done. Electron returns back to the original position. We call it as cyclic photophosphorylation. And when the electron do not return back while production of ATPs and other products, it is known as non-cyclic photophosphorylations. So we'll start with the first one, the more simpler one, that is cyclic photophosphorylation. Both are easy, but because cyclic photophosphorylation involves just one photosystem, it is quite simplified form of process. So let's see what this process is. So cyclic photophosphorylation. Photo means light. Phosphorylation means what? Phosphorylation means ATP formation, right? You all remember now. Cyclic photophosphorylation. Chalo. Here in this process or in this type of photophosphorylation, the ejected electron, when the electron is sent from the photosystem, suppose this is one photosystem, P700 of course, electron is ejected, but this electron will go through the electron carriers and will move back or will get returned to the photosystem again. So here a complete cycle is formed. Such kind of cycle is known as cyclic photophosphorylation. So we can say the first point, electron returns back. I'll just keep that picture only, you will think what I have drawn. Electron returns back to photosystem. Which kind of photosystem is used? Photosystem 1 is present. So cyclic photophosphorylation has only and only photosystem 1, that is chlorophyll A700 reaction center will be there, not chlorophyll A680. Next point, because here photosystem 2 is absent. Now, this forms important questions asked in exams already, because cyclic photophosphorylation do not have photosystem 2. What are the consequences or results? Here, no photolysis of water is occurring. No photolysis of water. Means what is not happening? The water molecule is not breaking by the help of light photons. Photo means light, lysis means breakdown. So breakdown of water molecule in the presence of light, it is not done in cyclic photophosphorylation because it does not have photosystem 2. And when no photolysis is done, what are the products not formed here? Oxygen is not formed in the process. No oxygen evolved. Now, we can make a complete link, right? And on this link only questions are asked. Photosystem 2 absence is reason of no photolysis, and no photolysis is reason for no oxygen evolution. Now, here again one other point is there, that is they do not have NADP reductase enzyme. They do not have NADP reductase enzyme on the stromal side, and if this enzyme is not there, what will be the result? NADPH2 is not formed. No NADPH2. No NADPH2 means oxygen is not formed, NADPH2 is not formed in cyclic photophosphorylation. Only one product is there from cyclic photophosphorylation, and that is ATP. How it is formed, I'll show you soon. So what is formed in cyclic photophosphorylation? ATP produced. Okay, other than ATP, no NADPH2, no oxygen evolution, nothing. Now, one more important point asked in exams, different exams, number of times. Primary electron acceptor. Yes, who is the primary electron acceptor? Of course, I will show you the complete cyclic photophosphorylation, but I want you to learn, I want you to remember this primary electron acceptor separately so that you won't get confused when you are writing the exam. The primary electron acceptor is an iron sulfur protein. Fe means iron, S stands for sulfur. So it is a iron sulfur protein or iron sulfur compound, let's say it to be more specific. In the terms in which it is asked, it is an iron sulfur compound. Okay, now Here only ATPs are formed. These ATPs are going to be used for the dark reaction. Let's see how this complete cyclic photophosphorylation actually occurs. So, to understand cyclic photophosphorylation, where it is occurring in the thylakoids. So, first what we need to draw? We need to draw a thylakoid. So, this is one thylakoid lumen. Okay. And I'm just drawing this thylakoid over here. This is one complete thylakoid. Okay. Now, here what happens? It is very odd, right? Let's say this is lumen and this is one complete thylakoid membrane. Okay, better than the before one. What is present in the cyclic photophosphorylation? Which photosystem? Photophosphorylation. Which pigment system is present? We know that in cyclic photophosphorylation, we just have photosystem 1. PS1 is present. PS1 is present means which chlorophyll will be present? Chlorophyll A700 will be present. Now, what happens? Sun is there or normal light source is there. The light will be falling on this photosystem. Light photons will be falling on the photosystem. That complete light harvesting complex is present here, trapping the light, sending to the reaction center as we discussed earlier in this video. Now, when the light is received, photons are received, what happens? Electron is ejected. Electron comes out and this electron is received by the primary electron acceptor from photosystem 1. The primary electron acceptor of photosystem 1 is iron sulfur compound FeS. From this iron sulfur compound, the electron will be transferred to the next electron carrier, or we can say the first electron carrier which is present in the cyclic photophosphorylation, that is ferredoxin. It is ferredoxin. Now, this ferredoxin is electron carrier, means it will carry electron with it and it is going to give its electron to the cytochrome complex. It is going to give its electrons to a cytochrome complex. Cytochrome B6, cytochrome F. So B6 and F I am writing directly. It is a cytochrome complex. Now, in this cytochrome complex, what happens? They do not carry the electrons separately. Sorry. Electrons were carried by ferredoxin, but cytochrome is a special one. He says that I don't want just electrons, I want more. So what does this chloroplast or this thylakoid offers? This thylakoid offers the protons from the stroma. Of course, if this is thylakoid, then the outer part is what? This outer part is stroma, right? So the thylakoid offers protons to cytochrome, which has this electron proton carrier in it, that is the plastoquinone, and it takes protons and electrons together, and then further what happens? The cytochrome is going to transfer electrons to the plastocyanin. PC means plastocyanin. Okay, and what happens to the protons? These protons are actually sent to the lumen. The protons are sent into the lumen. So just imagine you and your sibling are sitting together having a plate of, we can say, chips, each with equal number of chips in them. Now what happens? You are just continuously taking, let's say your sibling is stroma and you are the lumen. No, I'll take another color, yeah, this one. You are the lumen. So what you are doing? You're just taking the chips from the sibling of yours. So what will happen? You will have more chips. In the same way, the protons from the stroma are continuously sent into the lumen. So protons will increase in the lumen and they will decrease in the stroma. Now, once your sibling realizes that he or she has a very little number of chips in the plate, what he will start shouting that I want my chips back. But this time you are going to do some trading. You say that, of course, I'll give you the chips, but I want something in return. I want something in return, and he says that, okay, I'm going to give you something in return, and he gives you some energy. In the same way, the stroma asks the lumen to send back his protons. So lumen says, okay, I'll send the protons back to the stroma, but I want something in return, and that returning, we can say the tip is the ATP which is formed here. So when the protons are sent into the stroma, what is being produced? ATP is produced by this process. ATP is produced, ADP converts into ATP. When protons move out of the lumen into the stroma, this we are going to study in detail in chemiosmotic hypothesis, for which you will have to stay updated. You will be soon getting the video for chemiosmotic hypothesis also, where we discuss how the protons actually move, what number of protons form 1 ATP, everything we are going to study. But here you just need to remember this much. Now, what about the electron? Okay, proton did its work. They had the deal for the protons. But what about the electron? The electron said that now once the protons have done their work, we want to reach our home. So the electrons are sent back to photosystem 1. Electrons will be sent back to their home, that is photosystem 1. And you can see a complete cycle is formed. Hence we give the word cyclic photophosphorylation, because ATP is formed in presence of light, where electron is having a complete cycle. Various electron acceptors are there, electron and proton acceptors are there, lumen is there, which is having collection of protons sending back them to produce ATP, and this is all about the cyclic photophosphorylation. Very easy, very simple. Remember that plastocyanin gives electrons to photosystem 1. This point you need to remember. The same thing is going to happen for non-cyclic photophosphorylation also. Quite similarities are there, so I'm just telling you what you can remember actually. So what you are going to remember, plastocyanin gives electrons always to photosystem 1. Photosystem 1 always gives its electrons to ferredoxin through FES, through iron sulfur protein. Soon we'll see non-cyclic photophosphorylation, and you will get to see the same thing, that electrons are given to photosystem 1 by plastocyanin, and photosystem 1 is giving its electrons to ferredoxin through FES. So this is what you need to remember, and I hope it is already in your mind. So now let's move to the second type of light reaction, or second type of photophosphorylation, that is non-cyclic photophosphorylation. So let me write the heading first of all, that is non-cyclic photophosphorylation. Non-cyclic photophosphorylation. I hope now you all are confident with this light reaction topic, because we have almost done everything important for this part. Non-cyclic photophosphorylation. Now, what happens over here? The word itself tells that electron will not return back to its home. So, it is not forming a cycle. We call it as non-cyclic photophosphorylation. What happens here? So many electron carriers are there. They have their own electric potential. And if we place them on the electric potential scale, they form a Z-shaped structure. Hence, this non-cyclic photophosphorylation is also known as Z scheme. It is also known as Z scheme. Here, the electron does not return back. So, electron doesn't return. Next point. Coming to the types of photosystems which are active in this kind of photophosphorylation, both the types of photosystems are actually active. So both PS1 and PS2 present, both PS1 and PS2 are present in this kind of photophosphorylation. Electron does not return. Here what is going to happen? Because PS2 is present. PS2 is present, so what will happen? Photolysis of water will occur. What is going to happen? Photolysis of water. Means now water is going to break. So let me show you how this water breaks. Water, then we take blue color only to show water molecule. Water is H2O. Now break this H2O. So we break H2O into H plus, that is protons, plus electrons, plus oxygen atoms or oxygen molecules, right? So how does photolysis occur, where it occurs, which forms one important question I'll show you when I'll be discussing the cycle actually. But remember, photolysis of water involves formation of protons, electrons and oxygen molecules. Three kinds of elements or three kinds of ions are important for this photolysis of water. That is, number one, manganese. Second is calcium. And third one is chlorine. Remember these three elements which are used for photolysis of water. Now, as photolysis of water is occurring, what is going to evolve? Oxygen is evolved. Oxygen evolved. So, no PS2, no photolysis of water, no oxygen evolution, cyclic photophosphorylation. But in non-cyclic photophosphorylation, PS2 is present. So breaking of water is there, water is broken, so oxygen is evolved. Clear? Coming to the next point regarding this non-cyclic photophosphorylation, they have NADP reductase in the stroma site. NADP reductase present. If NADP reductase is present, what it is going to produce? It is going to produce NADPH2. What it is going to produce? NADPH2 is going to be produced by this enzyme present on the outer surface of the thylakoid membrane. Now, next point regarding non-cyclic photophosphorylation is that ATP is produced. Of course, they are going to produce ATP. Almost same process is used by them. ATP produced. Now, what happens over here? The the primary electron acceptor in non-cyclic photophosphorylation, which accepts electrons from photosystem 2, is pheophytin. So next point is primary electron acceptor of PS2. PS2 means directly we are talking about non-cyclic photophosphorylation. Okay, it is pheophytin. Here, this question is asked a number of times in the exams. Now we are going to see the non-cyclic photophosphorylation complete process, which is also quite similar to the one we saw. So here we have got this thylakoid lumen. I'll draw the lumen again. This is thylakoid lumen. No, it is not perfect, right? Okay. This time it is nice. Now, the thylakoid lumen will have the thylakoid membrane also. Okay, this is thylakoid membrane, almost fine. Now, what happens basically here on the inner side of thylakoid membrane? This is inner side. Let me show you. I'll darken the inner side with brown color, and I'll darken this is the inner side of the thylakoid membrane, and the outer side is pink. I'm darking. Okay, this is outer membrane of the outer part of the thylakoid membrane. So now if I say inner part or outer part, you must not get confused between the two. So on the inner part of the thylakoid membrane, PS2 is present. Means chlorophyll A 680 nanometer is present, the reaction center. What happens? This PS2 gives electron. What it is going to give? Electron when it gets photons of light. So light is falling on this PS2 like this, and this PS2 gives out its electron. As soon as the light falls, it gives its electrons like this. Now, it is going to give its electrons to a compound that is known as PO-phytin. To whom it is going to give? It is going to give the electrons to PO-phytin. Clear? Now, this PO-phytin will give the electrons further to the next electron carrier, that is plastoquinone. But now this plastoquinone is very, very smart, and he says that I'll not take just the electrons. I will not carry the electrons, but I want something more, and hence the thylakoids offers protons from the stroma. Remember, this is stromal side. We discussed in the last part, like the cyclic photophosphorylation, and before that also. So H plus from the stromal side will be given as a reward to plastoquinone to receive the electrons. Now, this plastoquinone will carry electrons and protons and will give them back to the next type of carriers, that is cytochrome complex. Cytochrome B6, cytochrome F complex is there. Clear? Now, once the cytochrome receives the electrons will be given to cytochrome B6F. But what about the proton? So, plastoquinone says that I am done with my job, I don't want protons anymore, and he sends these protons to the lumen of the thylakoid here. Right? Now, the electrons, what they will be doing? They will give the electron, means the cytochrome will give electrons to the next electron carrier, and that is plastocyanin. The electrons will be given to whom? The electrons will be given to plastocyanin. And now tell me, plastocyanin gives electrons to whom? Plastocyanin gives electrons to whom? I told you in the cyclic photophosphorylation. Yes, they will give the electrons to photosystem 1, which is having the reaction center chlorophyll A 700 nanometer wavelength. Clear? So PS1 gets electron from plastocyanin, and now tell me that PS1, photosystem 1, will give its electrons to whom? It will give its electrons to the iron sulfur compound, that is FeS, and FeS is going to give its electrons to ferredoxin right? Now, the electron has been given to ferredoxin, but ferredoxin says that I don't want to give electrons back to photosystem 2. So, what it does? It goes to NADP reductase enzyme. It goes to NADP reductase enzyme, and this NADP reductase enzyme is very smart. It says that give me your electrons. It asks the stroma, means stromal side, to give its protons. It says ferredoxin to give its electrons. It asks the stroma to give its protons. And what it forms in the result? It forms NADPH2 as a result. NADPH2 is formed as a result. Now tell me one thing, what is the site of formation of NADPH2? One important point. Stroma is the site for formation of NADPH2. As you can see, we have NADPH2 formed in the stroma site. Clear? So, protons are continuously used from the stroma either to send into the lumen or to form the NADPH2. Meanwhile, photosystem 2 gave its electron to photosystem 1. Photosystem 1 received the photons of the light and gave electrons to iron sulfur compound. So it got its, means photosystem 1 got its electron requirement satisfied. But what about this poor photosystem 2? It also needs electrons to get sufficient, we can say, to get its requirements fulfilled. So it asks water H2O to break. Photosystem 2 will ask H2O to break, so that as soon as it releases its electron, water is going to give its electron to the photosystem 2. So, lysis of water occurs, electron is released, electron is given to photosystem 2. Protons are released. These protons, what will be done? So, since the photolysis occurs on the inner part of thylakoid membrane, they send protons to the lumen, and the remaining part, that is oxygen, it is also sent into the lumen. This oxygen evolves in the form of oxygen gas, but the protons remains in the lumen and they will lead to the formation of ATP. So let me tell you how they are forming the ATP. So here we have got this one. Okay. This is again the path of trading between lumen and thylakoid. Lumen says, I have lots of protons with me. I got protons through plastoquinone, who took protons from stroma and transferred it to me. I got protons when water broke and gave me protons. So I'm getting protons continuously, but the stroma side is upset because it has to send protons to lumen also, and it is giving its protons to NADP reductase also. So now lumen and stroma does some trading, and stroma says that I will give you energy, you give me protons. So protons will move from their high concentration to their low concentration, that is from the lumen of the thylakoid to the stroma of the thylakoid. And in return, what is being produced? In return, they are producing the ATP. ATP is produced by this, we can say, trading between lumen and stroma from ADP. So you can see that NADPH2 and this ATP, they both are produced in the stroma side, the site of production of ATP, and the NADPH2 is stroma. But the another fact that oxygen is evolved in the lumen of the thylakoid and it is released in the form of oxygen gas out of the plant. Or for respiration, they'll be using this oxygen, but not for photosynthesis. Now, this ATP is present in the stroma. This NADPH2 is present in the stroma. And what is occurring in the stroma? What is occurring in the stroma? Dark reaction. It's very weird. Dark reaction is occurring in the stroma, which is using this NADPH2 and ATP as byproducts of products of light reaction. Clear? So this is all about the photosystems, the light reaction you have to study. Coming to the dark reaction, guys, we already have the videos for dark reaction. I'll provide the link in my description box in which the complete dark reaction is explained. The chemiosmotic hypothesis, which supports the formation of ATP through this trading between lumen and stroma. For now, you remember this word trading. We'll discuss in detail in the upcoming video where I'll be discussing about chemiosmotic hypothesis for ATP formation. So guys, if you like this video, please like the video, hit the like button and subscribe my channel, hit the bell button to get the notifications for my upcoming videos and share it with your friends. Write your views in the comment section below.